So welcome, everybody. Thank you so much for coming over this early in the day. And we have a really wonderful program for you, a whole very exciting day where we're going to cover a lot of developments since last year. And the committee, the organizing committee, I really want to thank Raf O'Halloran, Pranti, Kundu, and Paula Croxon that did a wonderful job in organizing this program today. Okay, so in our Brain Imaging Center, we adopt a translational approach to imaging brain disorders. And we go across imaging modality, across different disorders, translating between the work that is being done in the clinic to the work that is being done in basic neuroscience labs. And I was mentioning before some of the disorders that we look at, and we can also label a few here, but if you don't see the disorder of your choice, just let me know. And really, we're also adopting an RDoCy type of approach, so looking at the common substrates that underlie those disorders. We also translate between departments in Sinai, but we also encourage collegial type of approach across institutes. So we really bridge at the Brain Imaging Center, we bridge between the work that is being done at the Translational Molecular Institute that is directed by Zahi Fayad, who's going to give a few words of introduction later on, and the basic neuroscience work that is being done at the Friedman Brain Institute at Sinai. We are bridging across department. Our goal is really to understand the shared and distinct mechanisms across diseases and in health, and to really accelerate the development of large-scale gene brain behavior data sets, and to contribute to the understanding, to the research that is being done now that's really revolutionizing our understanding of the brain, in that we can amass huge databases together to look at all these type of disorders on a common type of scale. When we started 2 years ago, it was in October 2013, our model was the Human Connectome Project. And this is an NIH-funded effort to chart the macroconnectome and its variability in healthy adults. There are 1,200 adults in this project, 300 twin pairs and their non-twin siblings, cutting-edge neuroimaging, of course, extensive behavioral testing, blood samples for genotyping, and it's all being put together in a data that is freely available and user-friendly, using this informatics platform to enhance this type of studies. And this was to serve as a baseline for future studies of brain disorders. And this is where we come in. This type of Human Connectome project included only healthy individuals, while here at Sinai, we really have the privilege of actually focusing and zooming in on disorders, psychiatric, neurological, the whole gamut of brain disorders. So this is our, the show of basically the vision of the Brain Imaging Center. We have the infrastructure provided by TIMI. We have the Siemens 3T magnetome Skyra, the MR PET. We have the 7 Tesla. We have a mock scanner. We have a lot of support that is being provided. And here at BIC, basically, the imaging support infrastructure and everything I'm going to show you in a minute is provided by TIMI and by BIC. But then the PIs are responsible for this part of recruiting, scheduling, consenting, blood screen, neuroscreens, and behavioral type of testing. Okay, so what we did in the past few years is develop a common protocol. So anybody who's interested in doing neuroimaging can have this common protocol where we have a localizer for consistent orientation and slice prescription. We have structural images of the highest resolution. We have bold fMRI, highest spatial and temporal resolution, field maps for correction of distortion, and diffusion MRI for white matter track imaging. And you can see here some of the specifications, but the whole entire protocol takes about 35 minutes. We have it optimized for 2 coils, the 32-channel head coil and the 20-channel head neck coil. And we have optimized it to be HCP compatible, so with the highest resolution possible for our 3T scanner, Skyra. So in the past 2 years, people and BIC users have been adapting this common protocol. So a person comes over, they have their own, for example, fMRI task, but the first thing is that we recommend to use that type of BIC common protocol across labs, across PIs, so we can compare results. And this is in the past 2 years, we have scanned with this common protocol more than 600 subjects. And we have structural images on all of them and more than one. People many times come back. We have T2s a little bit lower than that, but it's still very impressive numbers, including resting state. So this represents this type of a big data approach that we would like to encourage, and people that would like to think about some questions, to compare between disorders, to look at healthy individuals. If you have your own type of disorder already in mind, but you are lacking, let's say, healthy controls with the same type of common imaging, then it is available for use. And of course, when we do share data, we need to think about it very carefully. It's something that we would like to encourage. First of all, it is definitely in accordance with the NIH directive. It is also collegial, but it's great for science, and especially for neuroimaging, where every one of our own individual projects is restricted based on sample size. But here we can put together this type of big data to look at specific questions. And recently, you can see in the literature already the advantage of putting those type of data sets together. You can see it across many different disorders, and some talks will highlight those type of analysis. So we will, of course, acknowledge data use. We will have some prohibitions, and we are developing this type of MOUs to do this. But the data is here, and the data is available, and it is growing. BIC also supports a lot of peripheral type of views, installation, optimization. We have, so for the tasks, we have a monitor, we have an eye tracker, headphones, we have response pads. We support a lot of the psychophys type of measurements. And we have also, we give support for HCP compatible tasks, but also project-specific tasks. All of this data then that is acquired on the SCARA 3T is then goes through a certain pipeline. We push it to radiology packs for reading, so every one of those structural scans are read for morphological abnormalities, gross morphological abnormalities. It's anonymized. It is then pushed to XNAT, which provides the study database, the archiving, and it is there where we post our pre-processing pipelines that PIs can use. It is also used for archiving, sharing, and data transfer, for data exploration. We also have a supercomputer where we can do some of the jobs, and level 1 pre-processing. Oops. Then we have our own TIMI and BIC servers who have grown quite much in their storage from last year. We use that for backup, to test new pipelines, and for other purposes, and then the investigators receive the data, and it's basically seamless for the PIs, the data from the SCARA to their own type of databases. So BIC provides that type of support. So in short, we are supporting cutting-edge optimized acquisition protocol, peripheral measures, analysis pipelines for fMRI, for diffusion MRI. We also provide support for advanced study design and analysis expertise. I'm going to mention NIPIPE here, and you can hear about it more in poster number 2 this afternoon. And we really are amassing this very, very rich and very unique neuroimaging corpus of data. It's not every institute that can really support this type of imaging in psychiatric and neuropsychiatric type of disorders. Okay, the thing I think what we like the most in some ways is development. And here we also have great advances this year. So we started to do pharmacological fMRI. So what you're doing a scan while a person is under an influence of a certain drug, and you can do it in a placebo-controlled way to look at the neural substrates of the effect of the drug on the brain. We also support real-time fMRI and real-time neurofeedback for different reasons. One, for example, to reduce craving in addiction, but there are many other examples, enhance self-control. We have spectroscopy. We have also a PET MRI that I'll give an example of. We have a high-field 70 imaging. I'll give a few examples for that. And non-human primate imaging. In the future, we would like to do some more. We would like to also develop real-time motion tracking and monitoring, real-time fMRI, but with EEG and ERP simultaneously, and with BCI, brain-computer interface neurofeedback techniques. We would like to do some sodium imaging, and we're waiting a coil that should get here I hope, very soon. And interleaved TMS fMRI, which we're not doing yet, but this is definitely something we would like to invest in. Okay, so let me give you just a few examples of those developments. So this is something that we expect by the end of this month, actually, so it's really very fast. And this is for real-time motion monitoring and correcting. We have this 3-camera system. It monitors a set of distinct and reflective 3 markers that is worn on the subject's head. So the head position is estimated from the position of these markers, and the scanner coordinate system is updated about this motion. And then you can correct this motion prospectively and in real time. And okay, so basically the reconstruction is naive to the motion. So this is something that will happen pretty soon. Okay, then I want the example of a MR PET study, and this is in collaboration between Nelly Alia Klein of the NARC Lab and Zahi Fayad, the TME director. And here you can use this scanner to simultaneously acquire images of, for example, here we focus on the carotid, so this is outside the brain, but using F18 fluorodeoxyglucose, we basically can image the carotid plaque inflammation in people who are at risk for cardiovascular disease and in people who have disorders like cocaine addiction, and they don't have any overt symptoms of cardiovascular risk. But you can see that their carotid is actually inflamed. There is a plaque inflammation. While at the same time, you can use this 3D dark blood MRI to look at the structure of the vasculature. And here is the lateral carotid, and you can see the increased wall area and thickness as indicators of plaque burden. And here you can see the lateral, but you can see also the bifurcation of the carotid. So this provides a basic type of vascular aging indicators for disorders where symptoms are not yet overt. And this is a wonderful type of combined approach to look at this type of carotid basically abnormality, and this is so important to then resting state functional connectivity, thinking about the blood that flows to the brain. But we can also use it for molecular imaging, and of course the PET-MR depends on the radioligand that you have. So if, for example, we're interested in dopamine receptor availability, we can use the F18 ligand falipride. And there we could look at fMRI, a task-related activity or resting state, while at the same time measuring something like dopamine receptor availability. We can also challenge it with something like methylphenidate that blocks the dopamine transporter. So it's a wonderful tool for neuropsychiatric type of disorders. We would like to also enhance and recommend that people think about the 7T. So we have the standard protocol for the 3T, but we are now developing also a standard protocol for the 7T. And we will have it in a very standard way, similarly to the 3T. We will also have advanced sequences and analysis that will together support this higher resolution and higher sensitivity studies here. And right now, we are already using it. We're using it in humans and non-human primates. The use is, of course, not as wide as for the 3T, but still, it is definitely getting up there, and every person or organism, animal that goes in, we scan them quite a lot, which is what you expect. And recently, these are now very recent development that we have installed, and it's now going to be very ready for doing task-related imaging, because we have the monitor, we have all the equipment that is needed for task-related scans. So just a few examples on the 70, and if anybody is interested about subfields of some regions or sub-regions, so here's an example of the hippocampus and the subfields of the hippocampus, structurally speaking, so just the gray matter. If that's your interest, then you definitely want to choose the 7T over the 3T. For example, if you're interested in mechanisms of learning and memory, of extinction, this is definitely the way to go. If you're also interested in diffusion weighted imaging at a very high resolution, that's again a 70. I would choose the 70 over the 3T. Here's a comparison of the fractional anisotropy maps, colored with the direction of the principal eigenvector of the diffusion tensor. And you can see at the 7 Tesla that, of course, you will scan longer, so you scan twice as long, but your resolution is 5 times as good, basically. You have the voxel volumes are 5 times smaller with the 70 over the 3T. So again, if you have specific hypotheses about specific white matter tracks, this is the way to go. Also, if you're interested in resting state connectivity and really the ability to look at those type of, for example, the default mode network at a very high precision. So here is a 1 subject, 2-second type of frame. And in that less than 2 seconds, you can separate the non-bold, after doing some denoising where you separate the non-bold from the bold effect, you can already see the default mode network in 1 subject, 1 frame. It's pretty amazing for temporal dynamic type of imaging. So if you want to look at networks and how they interact in time, so not only how they correlate in time, but actually which one comes first when people are engaged in just daydreaming or doing some other things. So here is another example where, okay, the default mode network in the resting state can be identified pretty robustly also with the 3T, but this is single subject data and showing you that beyond the default mode network, you can also identify subcortical networks. That here, this is something that you cannot see on the 3T. You will not be able to identify with such a precision those type of ventral striatum type of networks, dorsal striatum, thalamic networks. You will just see one big blob. But here it's really tracking the anatomy. It's pretty amazing. And this is single subject. So it's not something you can do in 3T. We also do some fancy stuff. So even when we still need to develop the ability to quantify some images, but the BIC can provide those services of quantification when it is relevant across disorders. So here's, for example, a susceptibility weighted imaging with showing you this increased iron, these darker areas in the basal ganglia of a cocaine-addicted individual, and this is indicative of an increased iron deposition that can indicate a lot of different processes, indicative of oxidative stress, basically, that are relevant to across disorders, dopaminergic type of disorders, not only drug addiction, but also Parkinson's, dystonia, schizophrenia, maybe inflammatory type of processes. So we will be able to basically measure this type of increased iron in interaction with dopamine neurotransmission that can be very relevant across all of these disorders after we do some quantification work. Okay, and I would like to also mention our non-human primate imaging work at 3T and at 7T. The anesthesia equipment for ventilation and monitoring a full range of physiological parameters is set. It's working. It's wonderful. We have also a custom-built phased array and transmit-receive coil with 4 and 8 channels, 4 parallel and multi-band capacity. Here's an example of the multi-echo, multi-band resting state fMRI with the resolution parameters for you to see. MP2 range is developed, and multi-band diffusion MRI. And this is a group work led very much by Paula Croxon, but also others. Right now, the analysis and processing pipelines are being developed, and also imaging of the awake animal with eye tracking is also being developed. So in general, hours of scanning, so BIC basically started in October 2013. I think Timmy opened about January 13, but it started, I think, to operate around May 13, if I remember. So this is before, so this is just the start. And then you can see the hours of scanning increase, the expected increase, and the continued increase the second year of BIC with the support of everybody. And really what we are all interested in is new grants, right? And again, you can see this type of increase in the number of brain imaging grants, indicative of the support and the possibilities that are at Sinai. I would like to mention the ABCD grant, the Adolescent Brain Cognitive Development Grant. It's a landmark study on substance use and adolescent brain development. It will scan 10,000 children beginning at the ages of 9 and 10, and will answer some very, very core questions about the impact of drug use on structure and function of the developing brain, the brain pathways, and their association with the risk for mental illness, and many, many different really questions. And I'm very happy that Sinai will take part in this study. We will scan here at BIC around 500 children. We will follow them up longitudinally, and we will have a new toy to report about next year, I hope. Okay, so for anybody who is not yet familiar with us, this is the BIC core personnel. You can get our input on any of your studies, on any questions, or on any type of project every Monday between 12 and 1, HES 10th floor. We really welcome your participation, your questions, raising up any type of issues. It's a wonderful time to meet and discuss things and to get our opinion. We have a big steering committee, a very dynamic and lively discussion on type of big picture type of questions, and administrative personnel who are essential to everything that we do here. I want to mention an additional big forum, which is the BIC users group that meets on alternate Tuesdays. We have a very wonderful schedule coming up. It's a time and place where you would like to present your work. It doesn't have to be a finalized type of study. It can be just some preliminary results to get everybody's opinion. So I urge you to contact us to schedule your presentation. And with that, I'm gonna stop this introduction and welcome you again to this wonderful day of a lot of extremely interesting talks. And I want to thank again the organizing committee and everybody else. It's a lot of people who helped organize this wonderful day. And I'm gonna invite Zahi Fayyad to give his introduction. Thank you.
Okay, this is a wonderful way to kickstart this great meeting. First, I want to congratulate Rita and the BIC group for doing such a great job in organizing this imaging center. And I also want to congratulate you on a wonderful program. I'm super excited to hear every talk here and to interact with a lot of you. I just want to mention a few things related to the Translational Molecular Imaging Institute. So the Institute really is here for you. We put together with obviously the investment from the school that has been extremely generous with the building here that we are part of, with a series of imaging equipment that continues to expand, hopefully. So we have everything that we can do from mouse to man, including small animal and large animal, as well as non-human primate. I think this is a fairly unique setup. There may be some others here and there, but I feel the aspect and the mission that we are pushing, it's definitely putting us on the map, and you can see progress in all the areas. We really emphasize a lot the aspect of multimodality imaging. Traditionally, in brain imaging, has been, especially, in neuropsychiatric diseases and neuroscience, or maybe neuropsychiatric disease had been mainly, in neurology, had been mainly focused on the magnetic resonance imaging. But as the technology has been evolving and the type of data that you can collect today seamlessly and very easily, one great example is a PET-MR scanner, where now you have a PET scanner and an MRI in the single setting, We've also heard from Rita about the advances that have been made, especially through the Human Connectome Project, in terms of the utilization of high gradients as well as 3T and 7T to do macro imaging and better mapping of the connectome. So I think this is a wonderful era, and I'm so excited, to be part of it at this stage, so we can integrate this type of data. We have great support by our faculty members, by our technical staff, and you have seen from Rita the type of progress that have been already made in such a short time. Just want to emphasize a few things. How is our work with the Brain Imaging Center? So as you've heard, Rita and her group are really the bridge between our institute as well as the Freedom Brain Institute. This is a very important strategic alliance, and I feel this is a very tight collaboration between the technical team, the basic science, and the people who are doing translation. We and BIC, are obviously our mission as the principal investigators and a group is to be a driver of innovation in imaging. So you are more competitive as you are applying for grants and to do incredible science. As you've heard, also BIC has, I think, done a wonderful job in organizing itself and in creating these forums where people can come in, pick people's brain, request certain things, and get support and tailored support. Maybe I should call it concierge type of service related to your work. I mean, we really pride ourselves into this. You can get acquisition support. There's a lot of work being done in image processing that's expanding incredibly and will continue to expand, in my opinion. So we really feel extremely happy to be proud to push this mission, and I can see a lot of success, continuing moving forward. We mentioned, a little bit some of that, just so you realize it's not only the scanners that are available here, such as the 3T Skyra, the PET-MR, the 7T, the CT scanner, the MR, simulator, but also this whole series and batteries of peripheral equipment, including the coils, the screens, the anesthesia support, so you could do these things in the scanner, the computer, and then we keep hoping to expand more and more on the investment. Every year we introduce, hundreds and plus thousands of dollars in terms of equipment on top of what we have done. And we are working with Rita and the leadership here to potentially introduce, most likely, the Prisma scanner in our space that is open for development. So I hope within this year we work together to try to introduce this. This is really an incredible, I would say, pathway for us, new pieces of equipment. We've heard a lot about the 7T. I think Rita showed incredible examples. This is the area for us that we hope to grow, and we are starting to see growth in this area. I don't know if I can announce this, but I think Preeti Balchandani from TIMI and neuroimaging, she got a very favorable score on a grant that was reviewed yesterday. This is 7T imaging, and we expect a lot of other grants, especially with the preliminary data that I have seen on these slides, to support and to continue that program. One thing to mention also, we've heard a lot about the human imaging, but also non-human primate, but we also support, and we're trying to expand a lot more also in the area of neuroimaging for micro imaging, I will say, both with 7T MRI Bruker scanner and 9.4, as well as optical imaging, ultrasound. This whole idea, again, of trying to do not only a single, maybe organ imaging, it was mentioned maybe with that PET project or the important project we have with Rita and Nelly in terms of looking at both vascular inflammation as well as brain. I'm also working with other colleagues such as James Monroe, Chuck Teng, and others here in the effect of stress on inflammation and the driver of the myocardial infarction of stroke. So I'm doing brain imaging as well as cardiovascular imaging. This is a theme now more and more apparent in many of the grants that we're starting to see, the systems biology approach, trying to sample multiple organ, multiple data points to better understand this whole disease aspect. So we're happy to work with you. We're trying to expand also this facility by introducing a micro PET scanner, hopefully soon, along with the Prisma again. So I hope these new pieces of equipment will become online in 2016. So I just want to mention a few things. We have our 6th annual TIMI Symposium. This is save the date, April 22nd. We've already lined up the speaker. We're very proud about them. Bruce Fischel from Harvard will talk about, and MGH will talk about, obviously, his work is doing in neuroimaging and image analysis. Mark Griswold, who has been the pioneer in many areas in MRI physics, fingerprinting, which is a very important technique today that is taking more and more expanded into not only the area of the body, but it will also eventually touch the brain very soon. So we'll hear from him. Debbie Lee from Cedars-Sinai, a leader in cardiovascular imaging, motion correction to do fast imaging techniques to image the heart. And nanomedicine, we have Anna Moore from MGH also talk about her work in drug delivery and molecular imaging. And Julie Price from the University of Pittsburgh doing a lot of work in 7T-MR and epilepsy and other applications. So this is going to be a fantastic day, along what BIC Day is going to be today. So we look forward to your continued support and participation for April 22nd. Chris Canestrasi will continue to update you on the progress there. And finally, this is just upcoming lectures related to imaging in general. So Harry Tsumpas, who's a visiting professor here with us, who's an expert in PET imaging and PET image reconstruction, will be visiting, and during his visit, October 23rd, will give a lecture, an update, and this is actually great for the students who want to learn a little bit more about about imaging physics. This is really going into PET image reconstruction. And then we also have a speaker coming in on a topic also related to PET-MR, an evolving area, and not only in body imaging, but also in the brain, to talk about what people have done so far. Okay, so with this, I actually want to, again, echo what Rita have said, welcome you here. We're very excited about our keynote speaker also, who I think, and the rest of the program, who Rita, I guess, will introduce. Thank you so much.
Thank you very much. This was really wonderful. It shows how BIC and TIMI are complementary, and all the amount of work that's being done in conjunction. So let me now introduce Dr. Wilson Compton. Just to say that Nora Volkow was not able to make it at the last minute. There was a Senate hearing or something unexpected or other that prevented her from coming over. But we're extremely privileged to have Wilson Compton here. He's a nationally known expert on the causes and prevention of drug abuse. He's the deputy director of NIDA. Let me just okay. Dr. Compton served as the director of NIDA's Division of Epidemiology, Services and Prevention Research since 2002. As division director, he manages a complex research program of a national and international scope addressing the extent and causes of drug abuse and the development of effective prevention strategies. He's working very directly with Nora Volkow for these purposes. Recently, he has been leading an effort jointly sponsored by NIDA and the FDA Center for Tobacco Products to field a large-scale longitudinal populational study to assess the impact of new tobacco regulations. And this landmark study is expected to include over 50,000 individuals, ages 12 and older. He has also been a member of the DSM-5 task force and the substance use disorders work group for the past 5 years. And he was, prior to joining NIDA, Dr. Compton was associate professor of psychiatry and director of the Master in Psychiatry Epidemiology program at WashU in St. Louis, as well as medical director of addiction services at Barnes-Jewish Hospital in St. Louis. Please welcome Dr. Wilson Compton, a clinician, scientist, and in the public health area. Thank you very much for joining us today.
Thanks very much. Well, thank you, and it's certainly a pleasure to be here this morning. I bring greetings from Nora Volkov. She was quite disappointed that she couldn't join you today, and spoke to me over the weekend and asked me if I would come join you all today. And I said, well, I certainly can't take your place, Nora, but I'll be happy to present some related information and bring a general talk about NIDA's efforts to this audience. I would not pretend to keep up with you all in terms of the neuroimaging and neuroscience that you all are conducting, but I'm looking forward to spending the day with you so I can add to my repertoire. Part of my responsibility as deputy director is to review every grant that comes forward for funding. So it's already exciting to me to see some of the names of people who I recognize on the program here. And I look forward to seeing every single name that I see on the program coming forward for funding. Frankly, by the time it gets to us, it's been through the hardest steps already. It doesn't mean that we always say yes, because we don't. But the hardest part is the peer review process. So hearing about somebody's great score, that does not assure funding, but that's a necessary and an absolutely the most difficult first step. The reason Dr. Volkov isn't here is that there's an appropriations hearing this morning in the Senate. And so what I'm starting with is a little bit of an update about our budget. And so we are glad that they want to talk to Nora. It's unusual, actually. Typically for a budget hearing, they want to talk to the head of NIH. So Francis Collins is, of course, the lead testifier, and we'll say probably 95% of the speeches. But they almost always include the very large institutes. So it's unusual for NIDA to be included, and I think it speaks, as far as we can tell, to the urgency of some of the issues that the addiction field is facing. And I'm going to share some of that with you because I think that is driving some of our current priorities, and I hope to see how your work will fit in and complement some of the sort of the major public health and population priorities that don't guide all of our work, because they don't. We take a long-term perspective and support a great deal of basic science work because we know that in the long run, that's where the great payoff will be. But we're always paying attention to the shifting landscape of drug addiction and drug abuse and need to be responsive to the current needs. This is just a quick snapshot of our current budget. We are hopeful that these numbers will be more likely. The year started October 1, so we don't know. We weren't sure that none of us were sure we'd be at work this week. Just a week and a half ago, we were all making plans to not come to work because when we don't have a budget pass, and if they truly decide not to pass a budget, we are on leave until they decide to appropriate the money to fund us directly. That doesn't stop research from ongoing because you all have grants that are already awarded, so you're able to continue your work, but it certainly slows us down and puts a major crimp in the review process. It's just a nightmare. So we're hopeful that that will be avoided. And we're certainly hopeful that some of the numbers that include a modest increase will be what we'll see. So that'll give us an opportunity to support the cutting edge work that you all represent. We have a broad portfolio. One of the key lessons here is there was a myth a number of years ago that all of our money was going into medication development. That's the purple part of this pie chart. And Nora highlights this to remind people that we spend an awful lot on our neuroscience program, both basic and clinical neuroscience. And it's not that we don't find the medication development to be hugely important, but it is not the majority of our funding. And so that was an important message at times. Now, I've got a couple other sort of organizational issues. Then we're going to talk some more about the major areas of science that we're supporting. That's the sort of structure for my talk today. We have just implemented a significant reorganization that does have an impact on the work that you all submit to NIDA. And I think it's in keeping with some of the themes, and I'll explain why. We had our advisory council, which is, for those who are not familiar, that's like a board of directors. It's a public meeting, and it includes a small group, 12 to 15 senior scientists and public officials that meet three times a year to review our grant portfolio. So it's both to provide a secondary review of grant submissions and also to take up a series of questions. And in this case, we asked them to review our Division of Clinical Neuroscience and Behavioral Research, that's DCNBR. where much of the work at the human laboratory work and human neuroimaging work would have been supported out of DCNBR to examine its structure and function. There have been some cutbacks in the number of staff. There have been retirements. Given the flat nature of our budget or shrinking nature of the NIH budget and our desire to put more funds into grant support rather than supporting our staff at NIDA, we found ourselves in the difficult position of not having enough staff in this division to really take care of all of their areas of responsibility. Given how essential translational neuroscience is to the overall mission, we decided to integrate our programs throughout the Institute so that we're taking a bit of a gamble here. Because when they're separated and fully supported by dedicated staff, we know that they will be well supported. But the goal here is to see, can we do as good a job in supporting the science and in fact maybe even provide some advantages by integrating particularly our clinical neuroscience with basic neuroscience. I thought it was wonderful to hear the example of how you all are linking animal studies with human studies. That's what we're trying to do. So if you had grants from NIDA, they were probably from our basic science division. Rita certainly had grants from our clinical neuroscience division. So now they will both be managed in the same division of neuroscience and behavior. I think this in the long run should be an improvement and will lead to greater translation, at least at that stage of translation where you're doing sort of basic to clinical integration. The Behavioral and Brain Development Program in DCNBR has been sort of divided into two parts. Some of that are population studies that are moved to the epidemiology and prevention branches, and some of them are basic mechanism studies to understand brain structure and function across development, and those are going into the new Division of Neuroscience and Behavior. The treatment development, which is behavioral treatment development, will be integrated with what had been our medications development division, and we've renamed it to highlight their expanded program, and it's now the Division of Therapeutics and Medical Consequences, formerly Division of Pharmacotherapies and Medical Consequences. You can tell it's a broader mission. It also includes brain stimulation studies, so as you all are thinking about some of these very novel, innovative approaches for magnetic stimulation of frontal lobes, and Tobansi and colleagues have been really emphasizing that from both animal and now some early clinical trials. That's very exciting. There's not much in this area. Anytime you hear a research administrator say we don't have much in the area, that's a clue that we'd like to see some more applications on a topic. We're also emphasizing what we're calling transdivisional research teams. These are work groups. One of the difficulties in any organization is even though NIDA is not that big, I mean, we're $1 billion a year, which that is a lot of money, but we're several hundred employees in our extramural that do the grant administration, and about the same in our internal research programs, the intramural research program. That's not a huge number of people. Despite that, you still get kind of isolated within your area of science. So if you focus on prevention, you kind of forget to look at the basic science folks. That was my responsibility, so I was well aware of that. And so what we are doing is developing more robust work groups and teams to build areas of science that cut across the different divisions. Whether these are long-standing groups like the genetics group or our HIV/AIDS program, but there are multiple work groups that we hope will lead to better integration, just like we asked you all to form full integrated transdisciplinary teams. We want our organization to do the same thing. We've had a couple of staffing changes, just to mention to you. I'm really thrilled that Dr. Carlos Blanco, who is at Columbia University and has collaborated with a number of you all at Mount Sinai, recently joined NIDA as the director of our Division of Epidemiology Services and Prevention Research. That's where I worked previously, and I'm so thrilled to have my esteemed colleague join us. It's wonderful for me personally as well as for the field. We have a new executive officer. You all might not know what an executive officer is. That's the person who makes everything work within our system. It's an essential job. It's the chief administrator for our organization. And since what we do is manage a large number of grants and contracts and a large staff, it's a huge responsibility, and we will be counting on Joellen to help us operate even more efficiently and effectively than we have so far. She hasn't quite started yet. She starts a week from Monday, so very soon. Okay, that's a quick view of a little bit about the budget. We're not super optimistic about major increases, but we're at least cautiously optimistic that we certainly won't see we don't expect to see a cut this year, and maybe a modest increase, which will allow us to continue some innovative new programs, as well as the major organizational change. I'm going to go through this as quickly as I can to save some time for discussion and questions. So if I'm talking fast, that's intentional. So let's talk a little bit about what's new across NIH. And perhaps most important to you all will be the BRAIN initiative, which did not get much money from Congress in 2015, but was supplemented. It actually was pretty much flat. And so, but it was supplemented by additional funds from the institutes that participate in the program. And we're hopeful for a significant increase. It's not meant to be our entire neuroscience program at all. Neuroscience at NIH is about a $5 billion program across all the institutes. So even if this is fully funded at $500 million, that's only about 10% of the overall budget. But because it emphasizes new methodology, new technologies, we think the payoff could be huge. And I think that is very relevant to what you all do here in terms of the development of cutting-edge technologies. So I know you all are paying attention to the funding announcements that come out from the BRAIN initiative. And if there's anything we can do to help you be more competitive, that is part of our job. I'm thrilled that our staff can work with the extramural scientists to help you be more competitive with your applications. And the reason we can do this, and it's legal and ethical and not a problem, is because review is kept at arm's length. So the folks managing and directing and implementing the peer review process are not part of that grant development world. We certainly won't write applications for you, but we're happy to give you suggestions and point you in perhaps directions that you hadn't quite thought about. We may be aware of where other people are going with their work before it hits the published literature. So it's a way to understand, to get some advice about sort of what the common themes are that are developing. BRAIN initiative is number one, and I thought for this audience that's the most important one. But the precision medicine initiative is the latest huge landmark initiative supported and developed by NIH. This is really Francis Collins' signature effort, and I see this given that he's entering probably his last year as the NIH director. Those are political appointments. There are two political appointments at NIH, the head of the overall NIH and the head of the Cancer Institute. All the other institute directors are civil service, so they are not political appointees and come and go based on career aspirations and success and other issues, but not based on who's elected. But given the interest in this initiative by Dr. Collins, there's been a tremendous focus on it at NIH, and the goal is to take advantage of innovations in electronic health records, in big data possibilities, to essentially collect a huge cohort. They expect it to be over 1 million volunteers. It may include already existing cohorts and new volunteers. Some of this I have to be a little vague about because they haven't figured out the protocols. It will be a little bit like the way BIC works. In other words, there will be a common core set of information that is required from everyone that participates in this, whether they are the new volunteers that come in sort of off the street and knock on our door in a virtual way because they'll probably be signing up online, but whether they're new volunteers or from existing cohorts at typically accountable care organizations, HMOs and the like. That they will have central common platforms, but there will also be opportunities for many detailed additional work. Even with a sample of 1 million, turns out that won't be enough to get at some of the details of clinical disease, but it is certainly large enough that they should be able to subset for many interesting combinations and over time accumulate very useful information about course outcome, and some unexpected findings in terms of correlations and potentially the mechanisms of disease onset and progression. This is, as you can imagine, a remarkably complex effort that includes, extreme examples of patient partnerships, use of as much as possible, electronic health records administrative sources, but it will include collection of biological samples for genetic studies. I don't think they've talked about neuroimaging as a major platform, but I am sure that that will be incorporated over time. And I think you all might be among those who push the envelope in terms of trying to incorporate them. I am quite sure that this medical center or others in the area will be involved in this partnership. It just got launched, theoretically. Given that we don't have a new budget, you can't launch a brand new huge program, but the goal is to start it this fiscal year. So as the budget unfolds, you will see more information. The acting director for the Precision Medicine Initiative is Josie Briggs, who's the institute director for the Institute for NICCH. It's the former Complementary and Alternative Medicines that has a new name that I'm blanking on. It's a temporary position because they will be doing a national search for somebody who can launch this. And they're going to need sort of two major areas of expertise. One is in understanding, establishing population science and cohorts, so that it's not just a million of sort of random folks, but how do we understand the characteristics of the volunteers for this study so that you can draw appropriate inference that can be applied to other populations, as well as the logistics of coordinating across many, many sites around the country and collecting the data in a coherent and useful way, and making it available to researchers for secondary analysis. That's the other theme I heard as you all, with the introductory remarks by Dr. Goldstein, is the utility that you all make of all of your data. And by making it available to one another and to the public, you really enhance the reach and the benefits of your science. There was a myth out there that other people would scoop us when we make our data available. It turns out that just doesn't happen. No one else is as fast with it or cares as much about it as you do yourself when it's your own project. So you are hugely motivated to get your major questions answered. And then you'll be pleasantly surprised that there's new information and new science that comes from the data that you worked incredibly hard to collect. So it turns out to be a win for everyone, including the original investigators. And we certainly expect the PMI to be part of that. There's a couple more what I would call core activities at the NIH that I want to highlight for you. One is this notice of the common rule, notice of proposed rulemaking. That's a mouthful. In order to change the rules that govern human subjects research, there has to be a full and open rulemaking process. This has tremendous implications for ethics, for how we take care of our patients. And it goes back to the Nuremberg Code shortly after the Second World War, given the egregious examples of human torture and testing prior to that. Multiple declarations over time. More recently, even in the 1970s, the Tuskegee experiments, an example of egregious unethical experiments. Seeing that there were issues around the use of prisoners in research and the startlingly unethical treatment of prisoners over even in relatively recent history. That's how the common rule that got issued in 1991 establishes the basis for our current system of IRBs that each institution has. Now the question is, how do these work, and what do we need to do to make these work more successfully for current science? Science now is much more likely to include multi-site studies, collection of data from dozens or hundreds of sites, difficulty with anonymizing data. As much as we like to think of genetic data as being confidential, it certainly isn't completely anonymous. It can't be, because it's your own genetic code. So if I know your genetic code, I can figure out which subject you were in a study. So how do we deal with that from a human subjects issue? There have been a whole series of meetings to help develop this update to the common rule, and please stay tuned. When you see the publication in the Federal Register, this is really important. Please read it and comment on it. It is a requirement in this formal process that we respond to at least think about and respond in a thoughtful way to every comment that's received. So that's why this is not just paying lip service to these changes, but it really matters. And it can help you do a better job in your research, but also make it safer for the patients and the public that want to participate in research. You all have heard a lot about the reproducibility and transparency of research findings, and in some ways, you're at the forefront already of attending to these issues. The large sample sizes, the focus on the proper statistics and the proper sampling design so that you are less likely to have irreproducible findings is some of the key features that you all already undertake in your training programs. Those are not universal, so those are some of the issues that we've seen. And this has been, I think, a great embarrassment to our field, how difficult it is to reproduce and replicate findings, even those that are published in remarkably major journals. And so paying attention to this is an ongoing effort for the NIH. There's a new web portal that was launched, and we're certainly focusing on training and incorporating principles of study design as one way of improving this in all of our training programs. Now, the last sort of common NIH thing I want to mention is a new program that Nora's helping to lead, which is to look at not just the epigenome and the genome, but to really look at the next step in what happens with protein synthesis and protein release. What are the post-translation modifications that can be understood? What are the tools we need to have to explore the relationship between the genome organization and its ultimate function? This is a really understudied, explored area. There are only a handful of examples of how these may work. Figuring out, since some of them are so short-lived, figuring out how to study this, both in vitro models as well as in vivo models, will be a remarkable challenge. So I'm excited, and I know Nora and the colleagues at NIDA, as well as across other institutes, are particularly excited to launch this effort. This will be part of the Common Fund efforts over the ensuing few years, so stay tuned. All right, so I've given you a quick update about some major NIH-wide issues, the BRAIN Initiative, the Precision Medicine Initiative, reproducibility, and just a snippet around the epitranscriptome work. Let's talk about some issues around NIDA itself. This is where I'm, of course, most familiar. There's been a major shift in our AIDS program. This is AIDS budget at NIDA. What percentage of our budget do you think is AIDS related? Do you guys know? Anybody? Hazard a guess. 5%, 10%, 100%? It's about 30% of our budget has to be allocated to AIDS research. So as the NIH is developing new guidelines and new overarching AIDS research priorities, that has a huge implication for NIDA. Anything that influences that huge a portion of your budget, and in some ways what it means is that the locus of control for managing this is shifting from institute priorities to NIH general. Now, some of this is in response to the notion that it is conceivable that the AIDS budget may be reduced somewhat, and so NIH is particularly keen to make sure that the core major areas across NIH for AIDS research and the seek towards a cure of AIDS will be supported. So the major themes that we are going to be supporting will be focusing on work towards a cure, reducing the incidence, including vaccines, new HIV therapies, and a bit around comorbidities and co-infections. There will be some cross-cutting work on basic research, health disparities, and training. There will also be a series of procedures for grants that might be considered part of the AIDS program. We don't quite know how these will work yet, and it's going to be complicated to implement. It means that if I think a grant is relevant to AIDS, I can't make that determination. Dr. Volkoff can't make that determination in isolation. We have to submit it for approval to the NIH. And then if they approve it, we can use AIDS money to fund it. If they don't, then we can still consider it under the rest of our budget. But that will increase the competitiveness for our non-AIDS budget even further. And it also means that it's quite possible that some of our AIDS budget will be transferred to other institutes that might have higher priority work over time. We've just launched a new council subcommittee to help us think through these issues and be very strategic. We think this is hugely important to the drug addiction field. What just happened in Indiana this year? We saw a remarkable outbreak of HIV, the largest single outbreak in this country, with rates of about 5% per year in that one town. Now, it's a small town, 4,200 people, with something like almost 200 cases discovered just in the last year. That's a remarkable onset of HIV and a rapid increase over time. It was driven by prescription opioid misuse, so it was driven by Opana use and injection. It had been preceded by some early warning signs in that there was an increase in hepatitis C, which is another infectious disease transmitted by risky needle use behavior, but it's much more transmissible than HIV, so it can possibly be an early warning sign. This reminds us of the essential nature of drug abuse research in reducing the onset and spread of HIV. And we hope that we will be allowed to continue this work because we think it is so important for public health and outcomes. Certainly, medication-assisted treatment is key in preventing HIV. That's a key strategy. We're focusing on new implementation, and I'll come back to this in a minute, where emergency departments are now, there's a wonderful study out of Yale showing that by starting heroin and other opioid addicts on buprenorphine in the emergency department, they could get much better engagement with treatment. It kind of was built on the observation that the ER docs were tired of seeing the same people over and over. They were referring them to treatment. They weren't going. They'd even sort of walk them down the hall, and it just wasn't successful. So they said, well, we're kind of the de facto primary care facility for these folks. Why don't we take care of them? And in that case, induct them on buprenorphine. And indeed, it shows much greater engagement in treatment and better drug use outcomes, at least so far short term. Now, can you do this places other than the Yale Emergency Department under the leadership of Gail D'Onofrio? I'm not sure, because she's a pretty amazing leader of this area. But at least gives us hope that it can be done, and we'll figure out the models and see if it can be done elsewhere. That might be one way to reduce HIV is by increasing the treatment, effective medication-assisted treatment for drug addiction. We're certainly focusing on new therapeutics for opioids, and this will come back to the other theme of the opioid epidemic that I'll highlight for you in a minute. Implantable buprenorphine is a system of administering buprenorphine that can last about 6 months. Actually just got an email this morning inviting me to a training session for clinicians on this. The company that is making these will have to train a whole cadre of doctors, and addiction psychiatrists and addiction specialists aren't typically that good at doing minor surgery in their office. So this will be a new procedure. It's like Depo-Provera. It's a little, short implantable devices, about an inch long, several of them that go in just under the skin. So it's very minor. But one of the advantages is an issue in drug treatment is if you need a medication every day, the drug user has to make a decision every day to remain in recovery and remain healthy. If I can do this only once every 6 months, that's only 2 decisions a year that can keep somebody healthy and moving in a positive direction for their lives. That's why this looks very promising to me, and we'll see if the FDA agrees that the data they've provided is sufficient for approval. It's under review right now. We support a series of individual level awards for the HIV area, as we do in a couple other areas. We, like NIH has Pioneer Awards, we launched a program called Avant-Garde a number of years ago for senior scientists with very novel approaches to addressing HIV. And we've launched a second program, which is for more mid-level and entry-level scientists called Avenir Awards. This just shows you an example of some of the projects that we're supporting there. All right, that was a little bit about our HIV program. I think you got a sense of why I pay so much attention to it and why it can be important. If you all have work that directly relates to HIV, what's going on with HIV in the central nervous system, how do we measure and examine the HIV reservoirs in the central nervous system, We need your help in figuring that out. One of the key pushes in developing a cure for HIV is to figure out where HIV is lurking. Even when you reduce the viral load in the bloodstream down to almost zero, it's still lurking in the immune systems, in lymphocytes, in various locations, including the central nervous system, where it may be particularly hard to penetrate with medications. So we need help in understanding how to image HIV in the central nervous system. And you all may be uniquely qualified to do that, both because of your skills in new methods and because of the patient populations here in New York that could help us with some of these key research questions. HIV was #1 research area. #2, prevention that we'll focus on. When we think about prevention, it cuts across the full range of science from the most basic genetics and epigenetics. After all, genetic risk will predict who will go on to develop a condition. That's essential for understanding and implementing prevention. We need to know who's at risk so that we can make sure that we get interventions to the right people. And so we have a similar entry-level individual award for genetics or epigenetics. Here's just 2 scientists who were recently awarded these. And these are open announcements, so pay attention to these if you're interested. Multiple RFAs in terms of both basic science and some applied sciences that apply to prevention. These slides are all here. You're welcome to them. I hope they'll be distributed to you all. You're welcome to recycle any of them. The taxpayer paid for the development of these slides. You're welcome to use them and certainly learn from them at your own leisure. Now, when I think about prevention, I'm going to highlight for you a couple of public health issues. One is something that's been fascinating over the last few years, which are electronic nicotine delivery systems, often called e-cigarettes. They certainly have both promise and real concern. I'll show you some of the concern, but the promise is obvious. I got an awful lot of people that can't quit their combustible tobacco use. Now in New York, you're very progressive with your using taxation and Clean Indoor Air Acts and others to reduce the obviousness of the tobacco epidemic. It's also been remarkable that as we've done a good job in addressing this epidemic in the general population, it becomes much more concentrated in psychiatric populations, whether those with addictive disorders or serious mental illness. In fact, there's been no decline in the rate of smoking in those populations in the last 15 years. I don't quite understand that because we actually have effective treatments, but I think part of that is that for the longest time, we didn't attend to it. The last place that you could smoke in most hospitals was usually on the psychiatric ward. Even in substance abuse treatment programs, they often scheduled smoking times where people would be given passes to go outside and smoke or go someplace else to use tobacco products. That's just an example of how we weren't sort of paying attention to what was killing our patients and taking advantage of the opportunities for intervention while we had somebody in front of us. Well, electronic cigarettes as a novel way to mimic the nicotine delivery that a combustible cigarette provides might be a way to help people who haven't responded to nicotine replacement treatments, haven't responded to varenicline, haven't responded to bupropion, that this could be a new way to reach some of those. But there's a real concern about new onset. And data that came out just last December showed us that in youth in this country, e-cigarettes are now more common than combustible cigarettes. That really surprised me. I was a little concerned that the data was incorrect because it was the first time this question was included in these surveys. But the CDC has now confirmed this with another large-scale adolescent study that was released a few months after these data from Michigan. Perhaps even more concerning because teenagers will try anything. The question is, maybe they all had already been using other forms of tobacco, either chewing or combustible. But it turns out that a significant number of the e-cigarette users had no prior exposure to either combustible or oral forms of tobacco. So this is a potential new onset for nicotine addiction. There's now been one follow-up study that's followed a cohort in California and shows that a significant number of the teenagers that start with e-cigarettes transition to combustible over time. So the concern about a new pathway into combustible tobacco addiction is at least it's certainly very concerning. The data is a little early, and so that's part of the reason we've launched this giant longitudinal study that Rita mentioned, is so we can understand the trajectories of tobacco use over time and look at, as FDA is now able to regulate some of these things, can they make a difference to improve the health outcomes?
How many use the cigarettes before the e-cigarettes came to market? Is it the same percentage? Because is it replacing or is it like
I'm not able to look at that in this study yet because they only looked at current use of e-cigarettes. So I can't look at prior use of e-cigarettes and then, in this study, I'm not able to look at the other way around.
epidemiologically weren't 20% of our 12th graders?
It's not, in the 12th graders, it's about 17, well, let's go back up here. This tells you how many use cigarettes. This is past month, so it's about 14% of 12th graders. That's actually the lowest level we've seen in this survey ever.
Yes.
So these are remarkable improvements compared to just 10 or 15 years ago. I think I answered it.
Yes.
Okay. Okay. First public health issue that I know Nora has been highlighting are the electronic delivery systems. They're now being used for marijuana delivery. They're now potentially being used for other drugs, but you see them on the internet and in some medical marijuana outlets that the vaporizer pens are being used. It's essentially a battery, a wire, and some kind of liquid. So the idea is the engineering can determine what temperature the wire will get to. For e-cigarettes, it's a glycol-based liquid, so it essentially has to boil to create a vapor that's inhaled. For marijuana, it may have to be a higher temperature, but it will vaporize lower than a combustible than combustion. So the potential is that there may be fewer harmful materials in these. But it's certainly a concern as there are new routes of administration for marijuana. The second major sort of public health issue that I'll be curious about how you all can help us with this, and I'll show you some early examples, is understanding marijuana and the brain. This is being driven by just the remarkable shift in the social and legal landscape. Most recently, Oregon just started selling marijuana in commercial sales facilities. But it's interesting. We have marijuana legalized for recreational use now in 5 jurisdictions. 4 of them allow commercial sales. Washington, D.C. is the 5th. It does not allow sales. So I don't have anybody studying this, but I'm curious whether Washington produces a different outcome. The law in Washington allows possession, use, growing, and sharing, but it doesn't allow any sale. So one of the obvious concerns in this development is a widespread commercial marketplace that's not been so great when it comes to tobacco or alcohol, that the profit incentives create an urge to find new customers and to increase sales. So that is certainly a concern when we have another habit-forming, potentially dangerous substance. And we haven't figured out how to regulate this very well for tobacco and alcohol, so I think the concerns around marijuana are quite obvious. Nevertheless, even though I think they look obvious, we need to study this, and we've been funding a number of people to help us understand what's happened in medical marijuana states and what's happening in these now recreational marijuana states. Some of the data that has surprised us is of the 20 million current users of marijuana, about 40% of them use it almost every day. That's about 8 million people in this country that are using marijuana most days. That's an awful lot of users.
Are they younger? Do you have the age?
The age group, I'm not going to show it to you here, but the age of use the increases have been primarily in the 18 to 25-year-olds and in the older population. The baby boomers are bringing some of their habits with them. So from the population that was exposed to drugs earlier in the 1970s, 80s, and 90s and is now hitting middle age have a much higher rate of drug use than previous older populations. It's not so much new onset as we see a cohort effect for that group. But we do see greater new onset for the 18 to 25-year-olds. I'll show you some young person data because this has surprised us a little bit. There's generally been an increase when you look at the global population, when you look at the rates across the country in the last 10 years. Here's the rates for the 12th graders. And one of the things that has surprised all of us is that while we've seen remarkable improvements in tobacco use by 12th graders, so that it's now at the lowest levels ever seen in our country, and alcohol has had a more gradual decline, but really a very positive decline over the last 15 years, and a remarkable decline. Marijuana has been increasing in the last 10 to 15 years. Not much. It's been flat for the last few years, but while that's been slightly increasing, the others have been going down. So we now see marijuana more commonly used by teens than tobacco. And that's a surprise to all of us that always think of nicotine as the first drug of abuse, first substance used. The potency of marijuana has been increasing as well, so some of the research that was conducted 20 years ago needs to be retested, given that the products being used out there in the community is quite different. Now, does it matter? This is some public health data would suggest that it does matter. These are 3 large-scale population studies from our colleagues in Australia and New Zealand, comparing those that never used marijuana during their teens versus those that used monthly, more than monthly, or daily. So it's sort of color-coded. And comparing the odds ratio for their outcomes 10 years later. So it's a longitudinal cohort studies looking at teenage marijuana use and mid-20s outcomes. They were significantly less likely to complete education, much more likely, this one seems a little bit circular to me. Of course, regular users are more likely to be cannabis dependent, but they also have problems with other drugs. And interestingly, and of great concern, there's also a much higher rate of suicide attempts in those who were regular marijuana users during their teens when they hit their mid-20s, suggesting a relationship to at least internalizing disorders. There's one study suggests a significant drop in IQ. These are difficult studies, and there are not very many examples of these. We don't have that many long-term longitudinal cohort studies that test IQ over time. There's some caveats to this finding. It's only among those who use regularly and consistently over many years. So those who only use during their teens or only later in life didn't seem to have this drop in their IQ. But it is a clinically meaningful drop, and that is not an uncommon clinical course for somebody to start during their teens and continue to use pretty heavily over the ensuing 20 years. Yeah. Like this, degree of exposure, especially to something that they call in London skunk, stinky marijuana. There tends to be very high potency, high THC, low CBD. There's been quite a bit written by colleagues out of London on particularly its association. First, they thought it was causal of all schizophrenia, but it looks like it's particularly relevant for an earlier onset of psychosis, In the US, we don't have that data, and there's some structural barriers to collecting that data. We have no research ability to collect samples, at least at a federal, using federal funding from local distributors in the different states. I think it's one of the unfortunate barriers to our regulatory system. In that it's keeping us from really important information about what people are using in those different places. We think volunteers would be happy to share small amounts of their marijuana with researchers for study, but we're not allowed to conduct that kind of work because purchasing and possessing is illegal. And the only marijuana that's allowed for research has to come from a farm that is the one licensed facility that NIDA supports out of the University of Mississippi. So that's a structural barrier to doing the exact kind of, I think, interesting and important work like you described. I want to focus a little bit on this association with psychosis. Nancy Andreasen first highlighted for this. this force a long time ago in a large study coming out of Sweden, suggesting that those who had used marijuana when they were conscripted into the army, it's amazing to me that they had that in their administrative database. It just tells you the value of some of the Scandinavian public health data system that's remarkable. And for those who had used marijuana more frequently, had a much higher onset of schizophrenia. That's certainly not causal, but it shows an association. It could be could be things that were unmeasured, and while the conscripts didn't have overt symptoms, or they wouldn't have been brought into the armed forces, they could have had premorbid symptoms. So you can't say much from this other than there's an association. The Denoising study found something similar in New Zealand, showing that early onset of cannabis use showed a greater risk of psychosis. There's been some association with particular genotype, and as was highlighted before, particularly with certain types of marijuana, the heavy, more potent and regular use of the particularly high potency forms seem to be associated with an earlier onset of schizophrenia. I've been intrigued by a new finding that colleagues using, again, Swedish data are looking at. This is Ken Kendler and the a group of researchers out of Sweden that use the registries to look at hospital diagnosis and the diagnoses of schizophrenia and cannabis abuse, looking at different degrees of relationship. So what they're trying to do is adjust for genetics. We know that schizophrenia runs in families. So is the association with marijuana use simply explained by genetic predisposition for schizophrenia? So they look at how the relationship of these two factors is seen in this study, and then extrapolate to if they were perfectly genetically aligned, because you can see the changes in the odds ratios depending on the degree of relatedness. So they extrapolate to as if they were monozygotic, and they still show a strong relationship of earlier marijuana diagnosis to later diagnosis for schizophrenia. So this is suggestive that marijuana may be meaningfully and potentially causally related from at least using epidemiological data. It is specific to cannabis. That's intriguing to me. So it wasn't some general drug use risk factor, because when they look at other drugs using the same methodology, they really don't see an association, possibly one for the stimulants, but they don't see it for opiate sedatives or hallucinogens. And one other public health finding, and then we're going to switch to a little bit of neuroscience. There is another twin study coming out of the Australian and WashU group. This is Pam Madden and Andy Heath at WashU, along with Australian colleagues, that looked at whether those that had the genetic risk using polygenic risk scores. So this is a broad measure of genetic risk for schizophrenia. And those that had more of the genetic risk were more likely to report marijuana use. So these folks would suggest that genetic risk predisposes to marijuana use as opposed to the other way around. So that just tells us how complicated this relationship may be. Now, we have seen a number of neuroimaging studies. They are frustratingly incomplete. So this is where you all can play a key role here. Some interesting work suggesting that the cannabis CB1 receptors are lower in marijuana users in many brain regions. Interesting work suggesting that two particular brain regions were smaller, and this sort of made intuitive sense because these are regions important for memory or emotional regulation. So if they're smaller, maybe they're not working as well. That, as a clinician, was an easy interpretation. But when we look at a much larger study that looked all across the range of projects and not just looking at one study's outcome, what they find is that we can no longer conclude that there are significant structural differences that are consistent across samples and studies. So this is a very important project that both had a large study in and of itself and looked at all the other studies in this of this and conclude that there are no consistent findings for structural differences. But there still may be functional differences in terms of connectivity. We see this coming out just a few years ago in terms of fimbria. differences for marijuana users versus not, depending on the age of the regular use. We see some, what I conclude to be complexity in long-term marijuana use versus controls coming out of the New Mexico group, showing first structural differences that I'm not putting too much into, but then some intriguing issues in terms of the connectivity and showing a change in terms of the years of usage. So first maybe a dip and then an increase in terms of changes depending on how long people had been using marijuana. That makes some sense from a clinical perspective in that it may be very important to look at the duration of exposure. That will be true for all sorts of substances. This brings us to why we've launched the ABCD study, that I'm so excited that you all will be contributing important data to this landmark project. The goal of this project is to conduct biennial imaging along with annual in-person assessments. We're not sure how frequently there will be web-based surveys. You do that for two reasons. One, to keep your subjects engaged in the study so you don't lose track of them. And also to collect useful, important information that might change more rapidly than yearly or every other year. We're starting prior to the major onset of any substance exposure, so starting with 9 and 10-year-olds. And there's sort of two key components to this study. One is the sampling. Who's going to be included in the study? How many of them are general population of low or average risk? And how many of them are at high risk? Now, high risk at that age will be based on behavioral determinants, so maybe family history, as well as the typical things you look for are externalizing symptoms. So difficulty with control of behavior is typically who are at higher risk of using substances a few years later. The study is just getting launched. The awards were just issued, I think, two weeks ago now, and the first meeting of the investigators will be just after the Society for Neuroscience, so it's in about 2 weeks in Chicago. This gives you a snapshot of the collaborators. I think I might have left off a couple of the additional sites where data is being collected, but these are all of the sites where the research hubs are funded. It's being coordinated out of the group at UC San Diego, both for the coordinating center and the data analysis. This was a complicated grant submission process. Applicants could come in either as part of a large consortium or they could come in independently. There were two different reviews. One review looked at the coordinating center and data analysis center, so sort of the overarching coordination, and then another review looked at the research sites. And most of the sites were part of a single consortium, so most of these sites are out of the consortium organized out of San Diego. But there are a few of them that come from the sites that provide additional populations of interest, whether that's the group in Hawaii that has a unique population and some really wonderful neuroimaging expertise, or the group in Florida that has access to a remarkable and an important Hispanic subpopulation that will make sure that we have a diverse group to study for this landmark longitudinal project. Just like your center wants to make data available, a key goal of this project will be to take those 10,000 subjects and their MRI data and make that readily available to researchers all around the country. So I think your example of how the BIC program has worked will be a wonderful example for how we can make this available, even including the ethics of how do you make data available without violating confidentiality and without putting the subjects at risk for their participation in the project. That was a long discussion about prevention, highlighting two major areas, electronic nicotine delivery and marijuana as emerging themes. Part of the reason for this adolescent study, the motivation, is to understand the impact of marijuana. But one of the elegant aspects of the ABCD study is that it won't be marijuana by itself, but in combination with alcohol and tobacco, which is what kids do. It's hardly ever that you find marijuana used, especially heavy regular users of marijuana rarely use it alone. And so that's going to be a complexity of conducting this study and trying to extrapolate for which is marijuana causing these outcomes, if any, versus alcohol and tobacco as the other two main substances you'll see. There may be a smattering of others too, but those would be the main ones. So let's focus on our third major priority area, which will include treatment. I'm going to wrap up pretty fast. I didn't realize how long I'd gone. I'm having fun talking to you. We're certainly working on medication development. I want to highlight for you one study that really intrigues me. It just was released last week. This is done in conjunction with the Food and Drug Administration's Tobacco Products Center. And it was a large experiment with about 800 subjects in 7 groups, a 10-site, multi-site trial that looked at what happens if we lower the amount of nicotine in tobacco products. My guess was if you lowered it a bit, people would compensate by smoking more. Although not all the researchers thought that, and I'm really pleased that they were right. We don't see compensation. We really don't see any benefit if you only reduce it a little bit. But the interesting thing is when you reduce nicotine a lot, which is within the potential regulatory authority of FDA now, we see a marked reduction in the number of cigarettes smoked. And in other measures, a decreased intensity of their nicotine dependence. So that gives us some opportunities. The last thing I'm going to highlight for you, and I'll go through this fast, is the opioid issues. This is a major public health concern. This is really why I think Nora got asked to join the Senate today, the Senate hearing, is because the public health, the publicity around the opioid epidemic has been remarkable. We've seen a huge increase in overdose deaths driven by the excess availability of prescription opioids. New York State's been at the forefront of using regulatory authority to change the way prescriptions are written. So in your emergency departments, you can no longer write for large-scale prescriptions for opioids. The maximum is 3 days in an emergency department, and that's a shift to reduce at least one route for excess availability. It's not just the prescription opioids. Heroin's been going up. Both use of heroin has been increasing significantly, and then heroin deaths have just skyrocketed in the last couple of years nationally, mostly driven by changes in the heroin marketplace. Heroin is now much more potent and much more widely available in parts of the country that never had it before. You have a long-standing heroin epidemic here in New York, or it's endemic here in many parts of the city. That wouldn't have been true in St. Louis, where I lived for many years. There was virtually none there in the 1990s. That's not true any longer. Suburban, rural areas have heroin that never used to have it. That's a real shift, and economic forces do drive behavior, and unfortunately we're seeing the outcomes. We are focusing on this in terms of changing prescribing practices, rescue systems in terms of naloxone, and increased access to medication-assisted treatment. I want to zip through some of this. And just remind you that research can help in terms of better approaches to pain. I was really intrigued by your idea of using MRI for feedback. Some of the early evidence was around using MRI for feedback around pain. So some of the pain signals could be reduced. I'm still astounded that individuals can use it in sort of a biofeedback mechanism for reducing pain. That hasn't panned out as a long-term treatment, but it's an interesting model for care. Might there be other approaches building on brain stimulation? Might we have TMS approaches or deep brain stimulation approaches for the treatment of pain that in that case wouldn't include exposure to opioids? These are at least some of the novel approaches that we'll be supporting, and so will other parts of NIH. Now, the last thing I'm going to mention for you is that we have a strategic planning process underway. This is a little bit arcane, but I encourage you to take a look. We are right now open for your comments, only till October 30th. So if you want to tell us what we ought to be focusing on, now's your chance. We've certainly highlighted, it's a very broad strategic plan. We are going to continue to support research across the full range of basic science, clinical neuroscience, applied research in terms of prevention and treatment development, and then implementation work. But we need everyone's help in figuring out within those what should be our major priorities. We can't afford to do everything. We'd like to, but we can't afford it. So we have to choose which major areas we will make our priorities, and that's what this is all about. And the last is a pitch for when you go to Society for Neuroscience, there's a day before the meeting begins on Friday the 16th. Any of you all planning on attending this? A few are nodding and a few raised their hands. Those of you who hadn't thought about it, go a day early. It's free, and it should be it's a joint effort with Alcohol Abuse and Alcoholism Institute. So it's now a joint addictions-focused day, and these are usually really interesting, wonderful symposia. They're widely attended. They typically have 700 to 1,000 people attend them. I'm amazed at how many stay an extra day, actually come a day early to go to this, but please join us. Thank you very much for your attention. I'm going to be here all day. I'd love to take a couple of burning questions now. I appreciated having some asked during the day, but are there key questions I can answer for you right now?
So let me ask you before opening to some more questions. Mentioning the ABCD study and focusing on that, which will allow us actually the Prisma. So having the high resolution scanning, like almost a 7T type of scanning, but in the comfort of a 3T type of scanner. So for that longitudinal large-scale study, we can already think about everything. We did not include the specific aims. So what would be your thoughts about the additional type of studies that can be used as add-ons to get maybe more into interventions, or studies that use imaging to test new interventions in this population? What would be your ideas on that?
Well, this is a population which is going to be most important for understanding the onset of drug use, so it's not going to be so relevant for the development of treatments. Maybe later on, but it's not going to be the 9 and 10-year-olds that teach us about treatment. But you might be able to look at some of the cofactors. I'd be very interested in how differences in the brain occur in relationship to attention deficit symptoms, occur in relationship to depression or anxiety that a large number of these kids will experience at 9 and 10 years old. So I would be thinking about co-occurring disorders that are already expressed at that age as something that could be very important to examine. You might find some ability to look at some novel treatment approaches when it comes to attentional issues, for example.
And real-time fMRI, for example.
I hadn't thought about that. It's a great question. This will be kind of like the way your BIC program works. There will be a common platform that's required of every site. And frankly, if that's all you do, we're thrilled. But each site will also add some of their own additional work to this study, and that's a wonderful opportunity for you all to add your own particular flavor and spin on this large-scale national effort.
Wonderful. Yeah.
So I have two questions. The first one relates to the preventive medicine, the personalized medicine approach.
Precision Medicine Initiative.
Okay. To the precision medicine approach. So in your description of it, is there an effort to include new data collection, or is it practically to develop algorithm to already analyze what's already available? This is the first question.
It's a combination of the two. So the idea is that some of it will include people who have long-standing databases and data records, and will take advantage of that. But this is meant to be a longitudinal study carried out over time with biological specimen collection on multiple time points. So there will be an opportunity for adding to the information that's already available. What I'm not quite sure is how they're going to combine new volunteers where I don't have electronic records on them. So there will be an assessment protocol for new volunteers that will include physical exam, history taking, and biological specimen collection. So it'll be a combination of, somebody going to New York Health and Hospitals system as part of that electronic health record versus new volunteers that come in because of their interest in a particular condition or wanting to be part of this effort.
And then can you clarify a little bit more the 4D nucleome, please?
Well, I can't clarify too much because it hasn't been launched yet. So it is a new approach but the idea is to help us look at beyond just the genetics. I mean, at some point we thought just uncovering the genes would be enough to tell us how diseases occurred and who was at risk. Turns out that hasn't worked nearly as well as we had hoped. It's worked. It's been a remarkable tool. but it has not been particularly useful when it comes to a lot of the more common chronic conditions that seem to have polygenic approaches. So what about epigenetics? How do we understand the influence of the environment? That's the next step, is what are there in terms of transcription variation. But when we think of the nucleosome and this epitranscriptome, we're thinking about post-transcription modifications. So what's going on with RNA, which can be very short-lived, that might be important in terms of how within the cell there can be variants and within disease and normal development. And don't push me too much further. I will refer you to the experts in this area for other thoughts. John Satterley is our local expert on this. He's been part of our the Epigenome Common Fund effort and has run that for NIDA for the last few years. And this is his new, he and Nora really pushed this, and it received widespread encouragement by the Common Fund program.
Okay,
Other questions?
so other questions? You'll be here with us throughout the day.
I'm going to
I
stick
hope.
out. Oh yeah, absolutely. I'm here for the day. I want to learn from you all. That's the benefit for me in coming here is a little bit to give you a talk, but that doesn't benefit me so much. But I appreciate the opportunity to learn what you all are doing and see the cutting edge work. I actually have a question for you. You talked about your elegant use of common data collection methods for your scanning. What about the common data collection methods for your phenotyping? Because if you don't have a good phenotype, the correlations and associations with neuroimaging are suspect at a minimum.
So this is our next step, is definitely to develop. We already have, in many of the subjects, we do have a very comprehensive set of variables we look at. We are very much interested in neuropsychological type of measures of cognitive function, emotion, personality. But we do have to homogenize it, and we already have a set of variables that we think are crucial and that we will start to apply across all these different protocols. Right now, we're not there yet, but maybe next year when you come back, we can show you some.
Love to hear that. Just like you require 35 minutes of baseline neuroimaging collection as the standard, and then you can do whatever else you want for your next 25 minutes or hour and 25 minutes, you might think about some of the same approaches for
Absolutely. So we have now a 1 hour, but to get then, it's a process to get everybody to do those type of assessments. Many people already do, and it is about resources, and we are definitely striving to get there.
What's interesting, we have our NIDA Genetics Consortium, which you would think would have exquisite phenotyping of addiction. Mm-hmm. Mostly, they only have commonalities at sort of the diagnostic level. And that's a problem, because we'd really like to be able to cut across different data sets and combine data at a much more fine-grained phenotyping. Yeah,
absolutely. I think it is a crucial element. Without it, neuroimaging data does not have the richness that it can have in terms of the interpretation. And comparing disorders can be done only if we look at the underlying core issues, for example, impulsivity that we would like to assess, right. But many, many different other traits. So we are going to get there. We will put that together. That's definitely on the list. And I think right now we're maybe ready for coffee, and then we'll come here back after coffee and cookies for our scientific part of the day. Thank you so much.
Thank you.